So I'm going to talk a little bit today about Friedreich's ataxia, which is an autosomal inherited disease. At the Jackson Laboratory, we have a mouse repository full of mouse models that are available to the scientific community to help with not only the research, but the advancement of various forms of disease. And certainly we're very concerned with all aspects of disease. You'll be hearing from Dr. Howell today about Alzheimer's disease, and certainly ALS and other very complex forms of diseases. But we also focus not on the very common diseases that we see within the aging community, but we also focus here at the Jackson Laboratory on rare diseases as well. Collectively, rare diseases have an incredibly negative impact, not only on our society, but of course individual patients. And so we're here to try to tackle a lot of these diseases as best we can. Today I'm going to talk a little bit about our efforts in Friedreich's ataxia. And this is a disease that I've been working with the rest of the scientific community on for the better part of the last 10 years. And it started working with FARA, the Friedreich's Ataxia Research Alliance, when Ron Bartek asked if we could, please help in collecting some of the mouse models for Friedreich's ataxia. And from there, we went on to study and develop some of our own models that I'm going to tell you about today. So just a little bit about Friedreich's ataxia to begin with. It's an autosomal recessive gene caused by a repeat expansion. And you'll see that in this diagram, the normal form of the disease, the normal allele, and I'm still trying to get my pointer. Not gonna cooperate. Oh, there it goes. So in the normal allelic state, there's about 20 repeats in the intron of the frataxin gene. In the repeat expansion, you can have anywhere from 66 repeats to 1,300 repeats, with increasing severity associated with the lowering of the frataxin amount. So it is characterized by a trinucleotide repeat expansion, and this leads to the reduction of the frataxin protein itself. So I think it's important to realize that Friedreich's ataxia is not a disease of no protein; it's a disease of low protein, and for that, it's a very attractive disease in terms of optimizing and thinking about disease-altering or disease-modifying strategies for this disease. Most of the patients with Friedreich's ataxia start to develop symptoms by the time they're 10 to 15 years old. It usually occurs with some clumsiness as a result of the ataxia that will progress over time. They do have problems with overall muscle weakness, scoliosis, fatigue, and there's also a high degree of diabetes associated with this particular disorder. A lot of the patients actually succumb to cardiac deficits because, as I'll describe, this is a mitochondrial disease. And so in addition to the neurological aspects and overall weakness as a result of the lowered frataxin levels, the cardiac, the heart is severely affected. Just advancing through these slides. So I'm going to talk a little bit about what happens in Friedreich's ataxia. And as I already mentioned, it's a mitochondrial disorder. And so on the right here is a depiction of a normal cell, and the mitochondria are shown here. And you have the activity of iron-sulfur clusters forming and normal activity of iron accumulation that's required for normal cellular function. But in Friedreich's ataxia, there is a problem with iron-sulfur cluster formation, and the iron starts to accumulate in the mitochondria, and this becomes what we call a situation of iron overload. And that accumulation of iron overload in the mitochondria leads to free radical generation and subsequently, cellular damage. And so, being a mitochondrial disease has its challenges for sure. But as I mentioned, the disease is a disease of low protein, not no protein. And so for that, there's a number of strategies for Friedreich's ataxia that have been that are being pursued and have been pursued over the last few years. One essentially is gene therapy. So if this is a disease of low protein, if you use some of the standard gene therapy vectors to replace frataxin, that is certainly one strategy. Another is frataxin stabilizers. And so the frataxin that is generated off of the expanded allele, could you stabilize that protein? Could you enhance that protein? And can you use recombinant protein replacement as a strategy to add frataxin back to these cells? Certainly regulators of transcription and RNA therapeutics, if this is a disease of low frataxin protein, if you could tickle, if you will, the upregulation of the RNA and perhaps make more protein coming off of the gene, then that would be also desirable. And then, of course, there's modulation of other pathways that are associated with frataxin. And some of the more common therapeutics associated with the decrease in the oxidative stress and mitochondrial function, and as I mentioned, some of the RNA therapeutics. So I think the good news for Friedreich's ataxia is that there's a lot of shots on goal. There's a number of different strategies that we can employ. And we really just need the mouse models to start looking at these. So the mouse models of Friedreich's ataxia have been both exceptional and challenging at the same time. We really want to understand the mouse model. We want to use the mouse models to understand the disease for sure. I think the patient population and natural history studies have given us quite a lot of insight into the disease progression in individuals with Friedreich's ataxia. But we also want these mouse models to be a source of preclinical testing tools with respect to the therapies that are out there. Could we possibly utilize them in a mouse model that mimics Friedreich's ataxia in such a way that we might be able to predict efficacy and safety? There are multiple mouse models of Friedreich's ataxia that exist. I certainly am not going to go over all of them today, but I am happy to say that through the generous contribution of the collaborators around the world, a number of those mouse models exist here at the Jackson Laboratory in multiple forms, and they are open to the research community to order and use in the advancement of the understanding of this disease. So I'm going to talk a little bit today about two mouse models, or two areas of mouse models where a lot of the work has focused on Friedreich's ataxia. And one is creating the repeat expansions in mice using the human gene. And so these are patient-derived BACs or YACs, where the gene with the repeat expansion has been isolated and then inserted into the mouse genome. And then we're also going to talk a little bit about the specific knockdowns of frataxin in mice. And I think that these are some of the more promising mouse models of Friedreich's ataxia, especially when we're looking at therapeutic strategies such as gene therapy or protein replacement. So I'm going to go through just a few of them today. We don't have a ton of time, but there also are a number of mouse models that have existed where individuals have tried to knock in the high repeat expansions into the mice. And that has been the work of Massimo Pandolfo and a number of individuals who really have spearheaded and pioneered this field. And I really urge you to go ahead and look at those. I'm not going to talk about those today, but just focusing on these two human BAC transgenics and then the floxed alleles that allow for gene therapy and protein replacement. So the first one I'm going to talk about is the mouse model from Mark Kook's lab. And Mark Kook was, again, another pioneer in mouse models of Friedreich's ataxia and the studying of Friedreich's ataxia in general. And what Mark did was he took a patient-derived yak and he injected it into the mouse. And then in combination with knocking that particular mouse frataxin level out, you can get a mouse model that expresses no mouse frataxin and only the repeat expansion associated with the human expanded allele. And so this was an interesting model at first. When Mark first made this mouse model, he ended up having two insertions back-to-back in tandem, which actually gave you more frataxin than you really wanted it to. And so again, this is the disease of low protein. And so if we're going to model it, we really want a mouse model that has lower frataxin. Over time, he got a recombination in the genome and was able to isolate only one transgene, and that was really good news for the population. And then what we found is that these repeats were generally unstable. We had to. This was a really nice collaboration with Mark and myself, where we were able to get some of the mouse models that had 200, 400 repeat expansions, and then we were able to selectively breed those within the Jackson laboratory. And these took a pretty big effort with large mouse colonies and large breeding that we had to invoke. But in the end, we started to see a lot of instability. And so when the repeats were about 200, 300, 400, they were relatively stable, but every once in a while we would get a jump in the repeats. And we selected for those breeders to such a point where we got animals with over 800 GAA repeats as a result of just selective breeding in this colony. I didn't say my slides is not working very well. So let me go here and do it. So what did that mean in terms of frataxin protein? Well, in the cerebellum and the heart, you can see, compared to 250, 600, the 800 repeats were much lower. In a non-repeat expansion, a control of just 6 GAA repeats, you'll see that the frataxin levels are relatively very high. The same is true for the heart. In a non-expanded back transgenic model, you'll see that the amount of frataxin in the heart is very high, but in the 250 to 600, and then a significant drop in the 800. So our strategy here was to try to find a mouse model with the lowest amount of frataxin that we could, because even the mouse models that Mark Kook had started with were relatively subtle. And while it's not, and this is just the succinate dehydrogenase activity, which is also a marker of mitochondrial function that we look at to determine activity. Unfortunately, I think that these mouse models are really good for studying increases in frataxin levels, but to date, we have not been able to really elicit a significant phenotype in these mice. There were reported some initial phenotypes of maybe some minor ataxia, maybe some minor deficits in the rotarod. We were not able to reproduce those initial findings. And through concerted efforts with Helene Puccio and my lab, we both independently looked at this model all the way up to 18 months of age with very intense, I will say, phenotypic components associated with them, everything from treadmills to forced running to exhaustion to cardiac and every kind of gait analysis that you could think of. And with all of the various parameters that we looked at, unfortunately, we just have not seen anything significant. And so while the levels of frataxin are very low in these mice, low levels of frataxin, seem to be very well tolerated in mice. So again, just to summarize, this particular mouse model is exceptionally good for looking at increasing therapeutics that can increase frataxin levels and stabilize frataxin levels, but the physiological outcomes of those deficits are not realized in this particular model. So this leads me to move on to a different mouse model that was first really explored and established by Helene Puccio in Strasbourg. And this was a mouse model that basically invokes a frataxin conditional allele. So if the mouse models we have with the very lowest levels of frataxin could not be used to establish a phenotype that resembles the patient population, how then could we possibly use the genetic engineering in the mouse models that we had? And I'm going to explain to you the rendition that we have, which is a floxed exon 2. Helene floxed exon 4. And for various reasons, we decided to go back to exon 2 just to look at RNA stabilization and actually what kind of levels were coming off of this particular floxed allele. and the presence of some of the Cre expressing alleles that we put in conjunction with this. So in addition to making the frataxin flox allele, we also made a knockout. And very much like Helene Puccio, we used a strategy of a flox allele with a knockout over a conditional to have tissue-specific depletion of frataxin in various tissues to really sort of see what we could look for a phenotype. And this is just a diagram of what that would look like. And so here is the frataxin homozygous flox allele. In addition, we have the heterozygous null allele with either a cardiac-specific Cre or a neuronal-specific parvalbumin-specific Cre. And I'll show you the results from both of those particular mouse models. And so in one case, we're depleting frataxin only from the skeletal heart muscle. And then in the other form, we're depleting frataxin in a mouse only from those parvalbumin-expressing neurons that primarily function also in the cerebellum. So what you'll see here is the cardiac-specific depletion, and we have a lot of data on this, and I won't show it all to you for the sake of time, but I will say that this is a very robust model in our hands. So when the flox allele, in addition to one allele as a knockout, is combined with the cardiac-specific Cre, we get cardiac frataxin deficiencies in the heart that really elicit quite a nice phenotype. And so at 10 weeks of age, we can measure the deficits in these mice with deficits in ejection fraction compared to the wild type. You can see here that there is a serious decline in heart function, and the cardiac myopathy is very well pronounced in these particular animals. These animals will succumb to the disease very, very shortly after life, around 11 or 12 weeks, maybe a little bit longer in this particular model as opposed to the parvalbumin model. And this is a really efficient model that we've used quite nicely for gene therapy and protein replacement. And we found that using this model in combination with those experimental therapeutics have given us really nice survival and extension of life and cardiac function in these mouse models. And then again is the parvalbumin Cre, and so this is the interneuron expressing Cre that we've knocked out. And as you can see in this particular model, we do see neurological deficits. And this is a neurological score just simply based on observations of the mouse, its overall movement, its level of paralysis, hind limb clasping, and various components of a visual inspection. But we also can see that there's a latency to fall in a rotarod, and these mice are quite quite disadvantaged in this particular task. So when you look at these mice, their neurological decline is quite obvious. The lifespan of these mice is quite compromised as well, and these mice will succumb to the disease in about 18 weeks of age. And so the neurological deficits are quite clear, and again, using gene therapy or gene replacement, this becomes a really nice model to look at the neurological components of the disease in terms of the population and what we might be able to expect from various gene therapy trials and studies. So with that, I'll stop and just give a little summary here of some of the things that I've showed you. Like with many areas of many therapeutic areas, new mouse models for Friedreich's ataxia are always on our mind. We have a very active rare and orphan disease program here at JAX, and so we're always looking at precision mouse models that can help us better model diseases. And in this case, for Friedreich's ataxia, I think we've had some successes, and I think we've also had, I wouldn't call them failures, but disappointments in terms of lowering frataxin levels in the mouse. And I think that's interesting because it does tell us a little bit more about the biology, and that we have a little bit more work to do. I've shown you some mouse models of tissue-specific ablation of frataxin and their utility in uses of protein replacement and gene therapy. And then the mouse models of human frataxin as well. These are the lower for taxon levels that don't necessarily give us the same phenotypes that we were looking for. But again, mice aren't human and human aren't mice, and why mice are able to tolerate low levels of frataxin, I think, is a question that we're still looking for. And also, there could be, some very specific effects of tissue-based expression in these human yak models. And taken together, the mouse models that we have, what we know about the disease, we're exceptionally interested in testing therapeutic strategies, and we welcome collaborations with both academic and industry partners to see if we can't take the mouse models that we have, take the disease-modifying strategies for therapeutics that others have, and advance the therapeutics for these patient populations. And so with that, I'll thank you and stop right there.